Autophagy, the cell's self-renewal and recycling process, is one of the most critical mechanisms for maintaining youthfulness at the cellular level. It allows cells to break down and reuse damaged proteins, malfunctioning mitochondria, and other dysfunctional components, essentially clearing the internal clutter that accumulates with wear and tear. Proper autophagic function is associated with longevity, resistance to disease, and enhanced cellular resilience. Yet, as we age, autophagy becomes less efficient. This has traditionally been seen as an inevitable part of aging, often described as a programmed decline in cellular maintenance. But a closer examination of how autophagy is regulated reveals a different, more actionable interpretation, that the drop in autophagy is not the result of an inborn failure, but rather the consequence of persistent amino acid insufficiency. In this view, aging is again better understood not as a disease process, but as a prolonged state of subtle malnutrition. Autophagy is tightly controlled by a set of nutrient-sensing pathways, particularly those involving the mTOR and AMPK systems. While mTOR is often discussed as a suppressor of autophagy in the presence of abundant nutrients, particularly amino acids, this has led to a misleading oversimplification that amino acids, especially from dietary protein, universally inhibit autophagy. In reality, the picture is more nuanced. While acute protein abundance may transiently suppress autophagy, chronic protein insufficiency impairs autophagic flux in a different, more destructive way, not by inhibiting it directly, but by starving the cell of the very building blocks needed for efficient recycling and renewal. Autophagy is not simply a cleanup operation. It is a remodeling process that requires energy, enzymatic activity, and the synthesis of new molecules to replace those being broken down. Many of these processes are amino acid dependent. For instance, the formation of autophagosomes, the vesicles that enclose cellular debris, requires adequate levels of membrane proteins, signaling peptides, and structural scaffolds, all of which depend on the availability of amino acids. Additionally, key enzymes involved in lysosomal function and the degradation of protein aggregates rely on a steady supply of essential and conditionally essential amino acids. If those substrates are lacking, autophagy stalls midway, unable to complete its regenerative cycle. This is precisely what we see in aging tissues with impaired autophagy, a buildup of partially degraded proteins, mitochondrial remnants, and lipofuscin, hallmarks of incomplete cellular cleanup. Rather than being signs of genetic programming toward decline, these can be interpreted as biochemical evidence that the cell is trying to renew itself, but lacks the resources to do so. Over time, this leads to increased oxidative stress, reduced metabolic efficiency, and heightened vulnerability to disease. But these are not inevitable. They are nutritional outcomes. Low protein intake, particularly chronic consumption of diets insufficient in key amino acids like leucine, lysine, and glutamine, is common in older adults. Factors such as reduced appetite, dental issues, poor digestion, and misguided dietary restrictions all contribute. Unfortunately, the conventional wisdom that protein needs decline with age has only exacerbated the problem. In truth, the opposite is more accurate. Older adults often need more high-quality protein to maintain not just muscle mass, but cellular housekeeping processes like autophagy. When amino acid intake is adequate, especially when coupled with physical activity or intermittent fasting to stimulate autophagic signaling, the system is far more capable of performing its essential cleanup tasks. Studies have shown that restoring protein intake in older individuals not only improves muscle synthesis, but also enhances mitochondrial quality, metabolic flexibility, and overall cellular health. These benefits are often attributed to the anti-aging effect of exercise or intermittent fasting, but they are just as much a result of improved nutrient availability for cellular maintenance. From this perspective, impaired autophagy is not a disease state, but a reflection of chronic undernourishment, specifically a deficit in the very materials cells need to take out their trash and build anew. Rather than viewing the decline in autophagy with age as an irreversible slide toward decay, we can instead see it as a process that is nutritionally constrained. And constraints can be lifted. The idea that less protein is better for longevity has been popularized in some circles, often due to simplistic interpretations of mTOR research in animals. But humans are not rodents in nutrient-rich lab environments. We are complex beings facing oxidative stress, metabolic inflammation, and decades of environmental insults. To maintain autophagy and renewal capacity in that context, we need to ensure robust amino acid support, not to overload the system, but to allow it to carry out its most basic, life-sustaining functions. In the end, autophagy is not a fragile function that simply fades with age. It is a living, dynamic system that depends on a constant input of materials and signals to operate properly. When that input falls short, especially in the form of amino acid insufficiency, the system slows and biological clutter accumulates. This is not aging by design. It is aging by deprivation. And that means it is not only understandable, it is, to a significant degree, preventable.